Hi, good morning. My name is Labrado, like you should introduce me. And my mentors were Emily Graham and Dr. Diane Nemrod. And we worked on the microbial extracellular enzyme activity and community assembly post-fire disturbance. Okay, so to introduce the topic, I want to start off with why do we care about microbes? So not only is the most diverse and abundant organisms on Earth, but they also control and regulate a lot of biogeochemical cycles. And so the cycle that we specifically focused on was the carbon cycle. And so as you can see, this is the carbon cycle. And what these microorganisms do is help in this area where it says decay organisms, and they help decompose organic matter. And so not only are they able to release CO2 into the atmosphere, which is we all know is a greenhouse gas, but they also make organic carbon available within the soil to other organisms. And so the way they do this is through extracellular enzyme activity. And so extracellular enzymes are secreted through microbes, and the two we focused on were NAG and BG. And so BG breaks down cellulose, whereas NAG breaks down chitin. And these two processes help in the degradation of organic matter. And so we wanted to analyze what the response would be to a burn disturbance. Burn disturbances usually are like a reset button on the environment. And so they kind of wipe out everything, and it's like a new, clean slate. So we wanted to see what was going on with that. And so in order to analyze what was happening after a fire disturbance, we also want to see what was going on with the community assembly. And so the relationship between EEA and community assembly is very important because structure can affect function. And so you want to know how this fire is affecting the relationship so that you can see what's going on with the carbon cycle. And so my three questions were, what is the relationship between edaphic factors, which are just your soil parameters, the extracellular enzyme activity, and the community structure? And how are they affected by fire disturbance? So we just wanted to make it relate to how this is going to be affecting not only the carbon cycle, but what's going on with the bigger ecosystem picture after fire. And so the fire that I studied specifically was the 4 Mile Canyon fire. This occurred in around Boulder, right outside in the 4 Mile Canyon. And so this is the extent, I don't know if you can see that well, but this is the extent of the fire. And it happened in September 2010. And it was actually one of the hottest fires that this area has ever experienced. It's one of the reasons it's taking a while for the environment to heal. And so to this day, it's kind of still in that healing phase. And so where we sampled was right here near the start of the fire at the mouth of the canyon. And so we did two plots. We did an unburned sampling plot, and we used burned sampling plots. And we just wanted to compare what was going on with unburned and burned. And so these pictures were taken this summer, this past month with my mentor when we went out. And as you can see in the unburned, it's nice and vegetated. The trees have leaves, they're growing, everything's really lush. Whereas if you turn over to the burned samples, the trees are dead. They're barren, they're still black and charred, and there is some vegetation on the bottom, which means it is in a healing process, but it's definitely not as lush as the unburned. And so for the sampling methods, there were two collection times. One was in October 2010, which was 4 weeks post-fire, and one was in January 2011, which was 16 weeks post-fire. And so we took 8 samples from each. October had burned plots only, whereas January had unburned and burned. And so I actually was able to sample this summer, but my samples will be used for future work, whereas I was using data from these samples. These collection times. And so for methods, there were quite a few. As with anything in biology, you do multiple steps. And so we started off with the edaphic factors. And once again, it's just the soil parameters. And we did soil moisture, pH, total carbon and nitrogen. And that was for all of the samples. But for the 16-week samples, we were actually able to get ammonia, dissolved organic carbon, dissolved organic nitrogen, as well as biomass. And so after we did the edaphic measurements, we went on to do DNA extraction and pyrosequencing. And what pyrosequencing does is isolates the 16S rRNA gene, and this is how you can tell the community's species diversity as well as its abundance. And so then after that, we did fluorometric assays. And this measures the extracellular enzymes, and it does this by actually measuring the fluorescence of the NAG or BG. And so lastly, we got all this data, put it together in lots of Excel spreadsheets, and did statistical analysis. And this was done through the R platform, and I conducted correlations, ANOVAs, and regression. And so these are some pictures of me actually doing these methods. This is me fumigating the samples with chloroform, which gives you a massive headache, and nobody tells you that until you do it. And so this actually lyses all the cells, and it stops all activity. So it's like taking a snapshot of what's going on in that environment then and there with these samples. And so from my results, we were able to measure, see the difference across treatments through ANOVAs. And this just tells us what factors are able to predict, well, adaptive factors are able to predict across the treatments because they're very varied. And so as you can see on the bottom, I have my treatments, which were the burned January 2011 and unburned. And then the in-between one was the burned October 2010. And so the two adaptive parameters that were significant were pH and carbon to nitrogen ratio. And so you can see in the pH that the burned samples have higher pH values on average, whereas the unburned didn't. And then when you go to your carbon-nitrogen ratio, you have lower values for the burned plots, whereas the unburned is higher. And so through the regressions, we were able to get adaphic models and microbial models, as you can see on the top. And so this was able to predict the EEA process that was going on. And so we did analyze BG and NAG, but BG didn't have any significant predictors for either burned October 2010 or burned 2011 for January. And so we just looked at the NAG. And so the predictors for October were microbial models, which was very different for the NAG predictors for January 2011, which were adaphic models. And so that just means that the NAG was predicted by the microbial activity, which was more random, whereas the January 2011 had adaphic models, which means that the soil parameters were more important in predicting the NAG activity. And so when we went on to look at the unburned January 2011, we were able to see that we got both adaphic and microbial models that were significant for BG, whereas we only had adaphic models predicting NAG. And so this just could be said that the microbial communities were being driven by soil parameters. And so for discussion, we just wanted to think about how these communities assemble. So microbial communities assemble in two ways. One is deterministic, which is niche-based. So it's relying directly on the interaction between their environment and these communities. And this is when you have significant adaphic models. So your soil parameters are significantly predicting your EEA, your extracellular enzyme activity. And the other one is stochastic, which is a neutral process. And so this is when you have significant microbial models. And this just tells us that it's acting more randomly. It's very decoupled from their environment. And so these are just some examples. And I chose this picture because it's like the tree giving life to the underground. And it's showing the direct interaction. Whereas this is like a dandelion. It's very random. It goes where it wants when the wind blows. And so this is our conceptual model. It's adapted from Fahrenberg et al. 2013. And it's just showing the shift between the assembly process and the EEA relationship. And so we go into October 2010, which, if you remember, I said had microbial models that were significantly predicting NAG. And so that's up here, it's stochastic, which is our neutral process. And so it changes from October to January 2011 burn, because it goes into significant adaphic models, which means that it was more of a deterministic assembly process. And so the interesting thing was that January 2011 unburned was also seemingly deterministic in assembly process with our models. And so that brings us to some conclusions. EEA did correlate to assembly processes, and this was demonstrated through those models that we were able to conduct. And the shift to stochastic to deterministic microbial community assembly processes was really interesting because you were able to see that shift through the NAG processes. And so one of the questions that remains is what's going to happen to these burned samples? Are they going to seemingly be deterministically assembling, like our January Unburned, which was not affected by fire? Or are they going to go into a more stochastic phase after the environment starts to heal some more? And so future work would just be a longer-term study because, after all, mine was constrained by time. We did 4-week and 16 weeks. And so possibly the samples that I did could easily be able to tell us these things. And so some acknowledgments. I'd like to thank my mentors. They were amazing the entire summer, and I had lots of them, so it was great. Unofficial, unofficial. And I had an awesome writing mentor, Kathy Kelsey, everyone at UNAVCO, and definitely my cohort, and Rolf and Peter. So thank you. Any questions?
Very nicely done. My question is just kind of a general one. What was the biggest challenge that you had? This looks really complex, and I don't know what your background was prior to coming, but what was your biggest challenge this summer?
Understanding all of the ecology and microbiology and everything. Yeah, definitely, because my background's more of geology. And so I do rocks. These things don't really have genes or anything like that. And so it was really interesting to have that learning curve. But like I said, my mentors were really great, and they gave me lots of material and everything to work with. So, yeah. Sure.
Can you reintroduce microbes into the affected environment in order to accelerate the carbon cycle or jumpstart?
You can, actually, but it would be more of that stochastic process, I would think, because there's several categories that fall under it, and one of them is dispersal. And so that means you can literally walk somewhere, pick up a microbe, and take it to that area completely on accident just with your shoe. And so microbes can travel that way, and they can restart the environment that way, but that would take a lot of just like putting microbes. No, a lot of times you don't. It's more of a random thing. But their existence in an environment can depend if they are assembling deterministically, which means it's based on their interaction with the soil, their environment.
Really nice job, Amanda. Your unburned plot, you said, was mainly deterministic. Is that right? Mm-hmm. Was there any evidence that there were also stochastic things going on in the unburned plot, or was it purely deterministic?
There could be, because there are models for NAG. Because NAG had only edaphic models, we thought that the edaphic predictors were probably driving that microbial community and their processes, and so it seemed to be more deterministic than stochastic.
Okay, but you did see some stochastic evidence with the NAG.
Yeah, we could.
Okay.
So that's why the longer-term study would be interesting to analyze that. Thank you.
In your slide, when you show your results, you have a plot showing statistical analysis. I was wondering, did you use a specific tool to manage that type of plot?
The box plot?
Mm-hmm.
Yes, I used R, and I did ANOVAs, and then they were able to give us these really pretty box plots after we were able to see which ones were significant and or not.
Oh, great, thank you.